What's poppin', guys? Welcome to the steady state summary. In this video, we are going to talk about different biomolecule structures and the bonds that hold them together, as well as the thermodynamics behind some of these interactions. On the left is an example of a peptide bond. This peptide bond in particular is bonding a cysteine shown in blue to an alanine shown in black. A peptide bond is formed when the amino end of an amino acid attacks the carboxyl terminus of another amino acid, resulting in the loss of a water molecule and a covalent bond between carbon and nitrogen. Peptide bonds are very stable in nature, and because of their partial double bond character and ability to do resonance, they also have extremely low reactivity. On the right is an example of a phosphodiester bond, shown in blue. The phosphodiester bond connects the 3' carbon of a nucleotide to the 5' carbon of another. This should sound familiar, as these types of bonds connect nucleotides together in DNA strands. Phosphodiester bonds are found on the outside of these DNA strands, with the hydrogen bonding across complementary strands found towards the interior. This should make sense, as a polar molecule such as a phosphate will have favorable interactions with water surrounding the DNA strand. On the left is an example of a phospholipid bilayer. The hydrophilic head of a phospholipid molecule faces either the interior or exterior of the cell, while the hydrophobic tails are found in between and are shielded from interactions with water and other polar molecules. Two things are important to keep in mind here. This setup is extremely favorable. The hydrophobic tails avoid less favorable interactions with water and other polar molecules either found inside or outside the cell, as well as more favorable interactions occur between other water molecules. It is important to note that favorability is relative to water-water interactions, and this helps to dictate a lot of biomolecule structure in biochemistry. This phospholipid bilayer provides a selective barrier for the cell to things such as proteins and ions. Also, notice that the hydrophilic tails are not actually covalently connected. This is because the interaction taking place between the phospholipid molecules are simply London dispersion forces, a very weak interaction we discussed in previous videos. However, due to the sheer number of London dispersion forces taking place inside this hydrophobic center, the membrane is held together. On the right is an example of a glycosidic bond that is found between monosaccharides. Although the specifics of this bond are not necessary for understanding biochemistry, it is important to keep in mind what sorts of processes may interfere with the bond. For example, a water molecule is likely to interact with this portion of a carbohydrate due to the highly polar bond between the two monosaccharides, as well as an amino acid that may be able to donate hydrogens, such as lysine or arginine. This will be important to keep in mind as we move into the specifics of biochemical reactions, but for now, this is something to simply think about, not just for glycosidic bonds, but for the other interactions shown in the video as well. This chart comes from your packet given out in class, and although the information inside the chart is important, understanding this from a general perspective with biomolecule structure in mind is what is most important. A negative delta G value indicates that a reaction is spontaneous, meaning that it is also favorable. Involved in this calculation is enthalpy, which deals with the internal energy of a system, while entropy is at the tail end, which in simple terms can be thought of as the increase or decrease in disorder of molecules and compounds involved in a reaction. From a general standpoint, think about the formation of a peptide bond between amino acids. Because two amino acids are coming together to form a chain of amino acids, this is leading to a decrease in the entropy of the surrounding environment. Meanwhile, the bond is absorbing energy to be formed, which leads to an increase in the internal energy. A positive enthalpy and a negative entropy means that this interaction is in fact non-spontaneous. Now consider hydrophobic substances in water, otherwise known as the hydrophobic effect. Nonpolar substances tend to cluster together in polar solvents rather than mix freely, with the former being much more favorable than the latter. The favorability of waters mixing with themselves would indicate a negative delta G value, meaning this interaction would occur spontaneously in many natural systems. A good shortcut to think about this is that favorable interactions occur spontaneously, while unfavorable interactions occur non-spontaneously. If you have any questions about this material, feel free to ask me, Dr. Bernstein, or any of the other TAs. Stay easy.